So thank you for the nice introduction. The experiment I will present was part of a real-time neurofeedback clinical trial. And we are first going to discuss what emotional working memory distraction is, how we measured it, what we found, and the relevance of our results. So once upon a time, emotions and cognitive processes had been viewed as largely separate. In the past decades, though, there is growing interest in understanding their intricate interrelations. Working memory involves the dynamic processes of temporarily encoding information from sensory input, maintaining that information, and being able to retrieve it when necessary. Emotions shape how we perceive the world. They can facilitate stronger memory consolidation, for example, after a traumatic event, but they can also disrupt cognitive processes like working memory. This impairment is actually termed working memory distraction. Sorry, it's not working? Okay. And emotional stimuli can decrease working memory performance by recruiting processing resources. What are, though, the distinct neural processes behind this effect? Well, investigating this question was the aim of our study. We based our first hypothesis on previous neuroscientific results that showed that when participants were distracted by an emotional stimulus, their amygdala, occipital cortex, and ventrolateral and dorsolateral prefrontal cortex showed increased activation compared to when they were distracted by an emotionally neutral stimulus. So that was the basis of our first hypothesis, and our second hypothesis was based on two competing theories explaining why emotional distraction causes decreased working memory performance. One theory suggests that perhaps cognitive processes are depleted from increased emotional processing during emotional distraction, whereas another theory suggests that overcompensation is the case. So we hypothesized that in the depletion theory, VLPFC and DLPFC would show increased activation in the retrieval stage of working memory, whereas in the overcompensation hypothesis, we expected to find increased activation. So decreased prefrontal activation, increased activation. This was a within-subject study that included 22 female participants, all diagnosed with borderline personality disorder, or BPD for short. People suffering from BPD show increased reactivity to emotional stimuli and have increased emotional distraction, which made this clinical sample ideal for our study purposes. So emotional distraction can be behaviorally assessed with a widely used emotional working memory task, EWMT, which has 3 distinct stages per trial. In the memoranda stage, 3 random letters are shown. Then during distraction, either emotional pictures or a cross is shown, which are the experimental and control conditions respectively. These are the emotional pictures or examples of them. And in the later stage of the EWMT, again, 3 letters are shown that in half of the trials include a probe that was also previously shown in the memoranda. So each stage of EWMT was jittered in duration so that we could distinguish between the different cognitive processes per stage. And the behavioral measures were accuracy in identifying the probe and reaction times. So in total, the EWMT lasted for 8 minutes, and there were 20 trials per condition. And given the EWMT structure, we used a rapid event-related fMRI design. So the behavioral results indicated that there were no differences in accuracy between the 2 conditions, but participants showed longer reaction times after viewing negative pictures. For the fMRI analysis, contrasts were modeled per subject based on the 3 distinct AWMT stages relative to baseline. And contrast between the probes following a negative emotional picture and the probes following the cross condition were also modeled. Whole brain one-sample t-tests were computed per contrast on the second-level analysis, and complementary small volume correction analysis were conducted based on the a priori regions of interest, the left VLPFC and left amygdala. So the fMRI results showed that indeed there was increased activation in the left amygdala, left VLPFC, and occipital cortex during emotional distraction. And also there was increased VLPFC and DLPFC activity during retrieval, offering some support for the overcompensation hypothesis. So combining these neuroimaging and behavioral measures facilitates a comprehensive insight into the interactions between emotional and cognitive processes. Nonetheless, our sample only included BPD patients, which limits our generalizability of the results. And future studies could use healthy control groups and other clinical populations, since emotional distraction and emotional regulation deficits are associated with various disorders. And our, I guess, take-home message is that emotional distraction can lead to overcompensation of executive resources, as indicated by increased VLPFC activation and reaction times. Thank you for your time.